All right, picture this. A disease is ripping through a poultry farm. The clock is ticking, right? For decades, figuring out what was causing it was this slow, super frustrating process. But what if you could see everything? I mean, every potential threat, viruses, bacteria, even brand new mutations, all at the same time. Well, today we're going to dig into the tech that's making that a reality, and it's totally changing the game for protecting our food supply and honestly, our global health.
I mean, that really gets to the heart of the problem for vets and scientists, doesn't it? How do you fight something you can't even see? When a whole flock gets sick, the cause could be anything. A virus we know about, a brand new variant, or heck, a whole cocktail of infections hitting at once. And every wrong guess, it costs time, it costs money, and it could cost the entire flock. You absolutely have to know what you're up against. And yeah, that's where it gets really tricky.
So for so long, we've been leaning on these classical diagnostic tools. But the thing is, they've got these massive blind spots. Think of it like this. They can only find something if you tell them exactly what to look for. If something new or unexpected pops up, they're completely blind. But now, a whole new way of thinking is taking over, one that promises to see everything all at once. And trust me, this isn't just a simple upgrade, it's a total revolution in how we track and fight diseases.
Okay, so before we jump into the amazing new solution, let's really get a handle on the problem itself. What exactly was wrong with the old guard? Why were these methods that we used for ages just not cutting it anymore?
Well, take a traditional test like RT-PCR. It's basically like asking a super specific yes or no question. You're asking, hey, does this bird have virus X? And you get a yes or a no. That's it. You don't get any of the juicy genetic details. And here's the kicker. If the real problem is virus Y, or something nobody's ever seen before, your test just says negative, and you're left completely in the dark, totally blind to what's really going on.
So yeah, those are some pretty big blind spots. Now, let's bring in the hero of our story. This is the technology that's here to completely rewrite the rule book on detection. We're talking about next-generation sequencing, or NGS, and at its core, this thing is an absolute powerhouse. You know the old saying, looking for a needle in a haystack? Well, forget that. NGS is like a machine that just vacuums up the entire haystack and gives you a perfect inventory of every single needle, every piece of hay, everything. It sequences millions and millions of DNA or RNA fragments all in parallel, all at once. And this is exactly why NGS is such a total breakthrough. It just smashes through the limitations of the old methods. Remember those blind spots? Gone. You can spot multiple viruses and bacteria in one go. You get the full genetic blueprint so you can track how they're mutating. And this is the real game changer. You can find pathogens you had no idea were even there. It's what we call agnostic detection. It doesn't care what it finds, it just finds everything. And here's the cool part. NGS is not a one-size-fits-all tool. You can think of it as having two main modes. On one hand, you've got targeted NGS. This is your sniper rifle. You know what you're looking for, and you want a super detailed deep dive. It's perfect for things like checking if a vaccine is holding up against a virus that's starting to change. Then you have the other mode, non-targeted NGS. This is like casting a massive fishing net. You don't have a specific hypothesis, you just want to see everything that's in the water. It's the ultimate tool for solving those "what on earth is killing these birds" mysteries. Okay, so this all sounds amazing in a lab, right? But you're probably wondering, is this actually being used out in the real world? And the answer is a huge yes. So let's take a look at how this is making a difference right now on the ground. Let's start with just one number, 785. What is that? Well, that's how many clinical samples were analyzed in a single massive study that put NGS to the test across an entire continent.
I mean, this was a huge project, a collaboration stretching across 8 different countries in Latin America. And using NGS, they essentially created a complete microbial map of the commercial flocks there. They didn't just find what they were looking for, they found brand new variants of infectious bronchitis virus, stuff they never would have seen with old methods. And maybe the most important part? By bundling samples together, a process called multiplexing, they brought the cost way down, proving that this super powerful tech could actually be affordable enough to use on a large scale.
Okay, this next part is really cool. What if you could basically do genetic time travel? Because that's one of the most incredible things about NGS. It can pull out secrets that have been locked away for years inside old tissue samples just sitting in a lab freezer somewhere.
So check this out. Researchers took samples that were 2, 5, even 7 years old, and they were able to reconstruct more than 99% of the viral genomes inside them.
That's incredible. It meant they could literally watch the evolution of something like Newcastle disease virus, tracking how it changed year by year. It proved that all these archives are basically a treasure trove, a gold mine of data just waiting to be analyzed.
All right, so we've established that NGS is this incredibly powerful tool. But having a powerful tool is one thing. To really make a difference on a global scale, you need to be able to use it fast. And you need to be able to use it on thousands of samples. You need scale. And that is where automation enters the picture.
In this right here, this is the secret sauce. What you're looking at are robotic liquid handlers. They take the single biggest bottleneck out of the equation: us, human hands. These machines can prep samples for sequencing with a level of speed and precision that a person just can't match. And they can do it 24/7.
And the impact is just massive. We're talking about going from processing maybe a few dozen samples to running 96 or even 384 all at the same time. This is what takes diagnostics from an animal-by-animal problem to a whole farm at once solution. Every sample gets treated exactly the same way, so the results are super reliable. Plus, the technology is so sensitive now that it works with just a tiny bit of genetic material from a simple swab. This is how you put detection on hyperspeed.
And it's not just about one robot doing one task. This whole thing is a beautifully integrated workflow. The robots handle the liquids and prepare everything. Then, hundreds of samples get a unique barcode so they can all be sequenced together in one batch. From there, the raw data shoots up to the cloud for automated analysis. And now, we're even using AI to sift through all that complex data to find patterns. It's a truly seamless pipeline, all the way from the field sample to the final answer.
So, with all this power at our fingertips, what's next? What does the future of flock health even look like when you have this kind of automated technology?
Well, this is where we see a really profound shift in thinking. For the longest time, the focus was just on identifying the bad guy, the pathogen. But now we can move beyond that. The future is about analyzing the host's response. We can look at the bird's own messenger RNA to understand how its body is reacting to, well, everything. It's like being able to ask, how is this flock feeling on a molecular level before a single one of them even looks sick?
And that ability opens up a future that is safer in some very real, tangible ways. Think about it. We could have truly precision vaccines, instantly knowing when a virus in the field has changed and our old vaccine won't work anymore. We could have digital biofoundries that could whip up a new custom mRNA vaccine for a specific flock in just a few weeks. And it even changes food safety. Imagine replacing slow, old-school bacterial culturing with a 4-hour NGS test that screens for salmonella and antibiotic resistance right at the meat processing plant. That's a whole new standard of safety.
And that really speaks to this expert view, right? The potential to conduct rapid and decentralized sequencing tests to detect pathogens will become more and more feasible as automation and new technologies emerge. Basically, the trend is clear. Everything is getting faster, cheaper, and closer to the source of the problem. What was once PhD-level research is becoming a practical, everyday tool.
So if there's one thing to take away from all this, it's this. Automation is the crucial bridge. It's what takes a brilliant technology out of the research lab and turns it into a powerful tool that can actually be used for global action. It smashes the bottlenecks and lets us, for the first time, see the entire microbial truth of a sample. And that just leaves you with one final, big question. Now that we have the power to see the entire microbial picture in a single sample, what's the next invisible threat that we're about to uncover? Because we're definitely going to find it.